Hello, today we'll be talking about MNase-Seq, which stands for micrococcal nuclease digestion with deep sequencing, and is a method to identify open chromatin. First, we'll talk about chromatin structure. Eukaryotic DNA is normally represented on a beads on a string manner, where double helix DNA is wrapped around 8 histone proteins. This protein DNA complex is called a nucleosome. This DNA is further compacted into chromatin fiber, and then this chromatin fiber is even further compacted into a chromosome. Next, we'll talk about chromatin accessibility. Open chromatin is DNA that is not wrapped around the histone proteins, while closed chromatin is DNA that is wrapped around the histone proteins. When DNA is tightly wrapped around the histones, it cannot be transcribed and then eventually translated into amino acids that form proteins. This makes knowing what chromatin is accessible to transcription is incredibly important to understanding overall gene regulation. Next, we will talk about the MNase-Seq method. First, our DNA of interest is treated with a non-specific micrococcal nuclease that digests naked DNA that is not bound to either the histone proteins or a transcription factor. DNA that is bound to either the histone proteins or a transcription factor is protected from this digestion. We are then left with DNA that is either wrapped around the histone proteins or bound to a transcription factor. Now, we might want to tell the difference between the DNA that is bound to these histone proteins or a transcription factor, and we do this by looking at the sequence lengths. The DNA that is bound to the transcription factor will have a much shorter sequence length than the DNA that is wrapped around the histone proteins. So, by looking at the sequence lengths, we can differentiate which DNA is bound to what. Once we have isolated the DNA from these histone proteins, we use a next-generation sequencing technique to get the sequence reads. This shows us what DNA was bound to the histone proteins and thus was unavailable for transcription and thus unavailable to be translated into amino acids and then eventually proteins. Next, we'll look at the method comparison. MNase-Seq is just one method to examine chromatin accessibility. There is also DNA-Seq, FAIR-Seq, and ATAC-Seq. As you can see in this graphic, MNase-Seq examines what DNA is bound to proteins. However, DNA-Seq and FAIR-Seq examine what chromatin is actually accessible. ATAC-Seq is a way to combine these two methods and attempt to examine both the bound and unbound DNA. Overall, MNase-Seq's ability to examine what chromatin is histone-bound and thus likely unavailable for transcription allows the researcher to get a glimpse into how these genes are regulated. Thank you for watching.